In this video, I'm going to be walking through the process of gluconeogenesis, where we're going to see two pyruvate molecules be converted back into a single molecule of glucose. And I'm going to start off by highlighting our bypass reactions, where there are three main bypass steps. The first of these is catalyzed by the enzyme pyruvate carboxylase. And so this carboxylase name shows me that we're going to have a carboxyl group that gets added to pyruvate. And so the way I like to remember that is that we have two molecules of CO2 coming in by way of bicarbonate. And so that CO2 is placed specifically in the active site by the biotin coenzyme. And we also need to recognize that carbon-carbon bonds are not going to be formed for free. So we're going to have this reaction that gets coupled to ATP being converted to two molecules of ADP and inorganic phosphate. So this is going to result in the production of two molecules of oxaloacetate, which has 4 carbons. First, I'm drawing the new carboxyl group that's been added, and then we're going to see the remainder of our pyruvate molecule, where now we have CH2, a carbonyl, and our carboxyl group at the end. So our overall transformation that we're seeing here is now we are adding this CO2 component to our original pyruvate starting material. And that results in us going from a 3-carbon species to a molecule that now has 4 carbons. And this molecule is called oxaloacetate. The second process for this bypass is going to involve the formation of phosphoenol pyruvate, also called PEP. And so we can abbreviate that because that's going to be useful in showing what's happening in our overall reaction here. And so just as a reminder, you don't want to just show the abbreviation, but because we have the full name here, we can show that the abbreviation is PEP, and so we can then name our enzyme PEP carboxykinase. And there are a couple of components of this name that we can find useful. We see carboxy again, and we see kinase that we've seen before. The carboxy is a clue that we're getting rid of that CO2 that we've just added, which makes sense because we have a 4-carbon species and phosphoenol pyruvate only has 3 carbons. So we need to get rid of that CO2 somewhere. But carbon-carbon bond breaking also isn't for free energy-wise. And so this kinase component of our name is telling us that we're going to have phosphate groups moving around, specifically phosphorylation, to make our phosphoenol pyruvate, where we'll see the phosphoryl group at carbon 2. And so this is facilitated by the consumption of 2 molecules of either GTP or 2 molecules of ATP. And the identity of the NTP depends on whether we're looking at this reaction being carried out in the cytosol in the case of ATP or the mitochondria in the case of GTP. And so what we're going to see is that this is going to produce 2 molecules of GDP or 2 molecules of ADP. And you can show both, you could show either one, as long as you show whatever source of the phosphate is, that's appropriate for your work. So that is our overall first bypass reaction, where now we have our PEP that has the loss of our carboxyl group and now the addition of our phosphoryl group. Our second bypass step is going to bring us back to the preparatory phase of glycolysis, where we're going to look at this phosphofructokinase 1 reaction, where we started off with fructose 6-phosphate and went to fructose 1,6-bisphosphate by kinase activity. To reverse a kinase reaction, we're going to see a phosphatase activity. And in this case, the phosphatase is called fructose 1,6-bisphosphatase. So just borrowing the name of our starting material here, fructose 1,6-bisphosphate, essentially there, and the phosphatase component of our name is telling us that we are moving and losing a phosphoryl group. So overall, what we're going to see here is that we are removing our phosphoryl group at the 1 position and replacing that with an alcohol that is sourced from a molecule of water. Remember, now we're into our 6-carbon species, so we don't need to think about 2 molecules of each of these cofactors or coenzymes or reaction byproducts. So we'll see 1 molecule of water come in to ultimately facilitate the release of this phosphoryl group. And so overall, that water is present so that we can have a source for our alcohol formation as we make fructose 6-phosphate. Our last bypass reaction is going to be the reversal of step 1 of glycolysis during reaction 11 of gluconeogenesis, where we're going to see glucose 6-phosphatase act on glucose 6-phosphate in the same way that we saw our bisphosphatase reaction. We'll see water come in as our supply for our alcohol at the 6th position. We'll also see the release of the phosphoryl group here in the form of inorganic phosphate. And so if we take a look at an overview of our process here, the only reactions that require bypass are reaction 10 of glycolysis here with pyruvate kinase, and that is facilitated by these 2 steps. We also see reaction 3 of glycolysis being reversed by step 9 of gluconeogenesis in our second bypass reaction. And in our third bypass reaction, we see the first step of glycolysis being reversed by step 11 of gluconeogenesis. But these are not the only changes that occur during gluconeogenesis. These are just the only ones that require different enzymes. And so for the remaining changes, we're just going to show the reverse of our components, or like our coenzymes, cofactors, byproducts that we had during glycolysis. So here by enolase, we had 2 molecules of water that were released. Now we're going to consume 2 molecules of water to go from PEP to 2-phosphoglycerate. If there are reactions that didn't require any cofactors, coenzymes, etc., we don't have to make any adjustment to those. So we're just drawing the arrow in reverse here, and we'll show 2 molecules of ATP are consumed to make 2 molecules of ADP here in order for us to go from 3-phosphoglycerate to 1,3-bisphosphoglycerate. And I'm just going to do the same for the remaining reactions. So glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, is going to produce 2 molecules of NAD by consuming our 2 molecules of NADH and protons. And that is our last step. Everything else is good to go. So that is our gluconeogenesis pathway.